Hello students, welcome to the EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute, commonly known as ICAR-NDRI, an institute located in Karnal within the green state of Haryana near Delhi, the capital of India. Today, I am going to talk about the module Sequence Alignment, Meaning and Significance from the paper Biostatistics and Bioinformatics. Friends, genes and proteins are linear sequences of nucleotides and amino acids respectively. The genes are passed on to the offspring in the next generations after replication of the DNA. However, during replication, the gene sequence, that is the DNA, may undergo mutation. But next generation offspring will survive only if they get a functional copy of the essential genes. If an essential gene is non-functional, then harboring organism, that is the offspring, will be wiped out of the population. With the result, mutated non-functional copy of the gene is also wiped out. Consequently, all the surviving individuals have at least one copy of each functional gene. The purpose of creating sequence alignment is to infer evolutionary homology so that the structural and functional information and knowledge of evolutionary related, that is homologous sequences, is used for modeling biological processes. In this module, we learn the biochemical meaning and the concept of creating protein and nucleic acid sequence alignments to delineate the residue-residue conservation during evolution. We'll then score the created sequence alignment to measure pairwise sequence similarity to draw an inference of homology. We'll also learn the visual presentation of the sequence alignment and distinguish among different types of alignments. During evolution, two types of mutations may occur: individual site mutations and complete gene duplications. Site mutations in genes happen during mistakes in replication, resulting in changed sequence which may change the structure of the protein and consequently the function to result in a different phenotype. Site mutations include three cases. First being substitution of one residue with another. Second is insertion of a residue in the sequence. And the third is deletion of a residue from the sequence. To understand site mutations, let us say that we have a protein sequence in one organism represented as sequence 1, SCQ1 in this slide. This organism acts as a parent and passes this sequence to its offspring without any mutation. Then mutations takes place during replication to result in changed sequence SCQ2 label. This mutated sequence is passed on to another offspring. In the present example, three substitutions are shown in red. First is the replacement of serine and threonine and other two are replacements of arginine and asparagines. The deletions and insertions of the residue in the first sequence with reference to the second sequence are highlighted using a dash symbol shown in black for several residues. Now let us understand the meaning of homologous sequences. The alpha globin sequences in different individuals, say frog, chick and mouse, inherited from a common ancestor 1 with alpha globin gene are called orthologous sequences. Similarly, beta globin sequences in different individuals, again in frog, chick and mouse, say, inherited from a common ancestor 2 with beta globin gene are also called orthologous sequences. Orthologous genes are called orthologs and are present in different individuals or different species. When these proteins are purified from these surviving and functional individuals and their amino acid sequences are determined using experimental methods, these two sequences will have identical residues on functionally relevant and significant positions, and the sequences inherited by each different organism from a common ancestor continue to perform same oxygen binding function. Its second type of mutation may also occur in an individual. This is called gene duplication. These are present in the same individual because they arose within a single organism by gene duplication. These duplicated genes, globin genes in the present case, in the same individual evolve independent of each other and may continue to perform the same function but maybe with different kinetic parameters. These chains are named as alpha globin and beta globin in the same organism and are different forms of the same protein within one single organism that bind oxygen but differ slightly in sequence and consequently in structure. This results in differences in their Km values as well as regulatory properties. These different forms of the similar proteins are known as paralogous sequences. The orthologous and paralogous sequences are collectively known as homologous sequences. It is the protein sequence which has encoded information for adopting a particular three-dimensional structure. The homologous sequences has several identical residues. Therefore, both might be having similar structure and consequently both might be having similar function. If the structural and functional knowledge is known for a protein in one of the species, then the same knowledge can be used for understanding the unknown structure and function of the same protein or the similar protein in other species. The sequences in different organisms can be compared for sequence similarity to infer homology and consequently to infer structure and finally the function. The sequences are compared through sequence alignment of similar residues in two given sequences. To create a sequence alignment of identical residues, let us have two sequences, with the first sequence having the length of 38 residues and the second sequence having length of 32 residues. The amino acid residues in the present sequences in this slide are not aligned. To delineate the identical and mutated residues in two homologous sequences, we need to create a sequence alignment between two sequences. To create a sequence alignment, each sequence is written in successive rows and each residue along each sequence in each row is written in successive columns. Then the identical residues from left to right are matched to determine the conserved positions along sequences in each column. To achieve this, for two sequences, the first residue in the first sequence in the first column and the first residue in the second sequence in the first column are compared, and if two residues are identical at this position in each sequence, then they are placed in the same column. If the first residue in both the sequences is not identical, then the second residue in second sequence is compared with the first residue in the first sequence. If they are identical, then first residue in first sequence is placed in column number 2 in first row, and second residue in second sequence is placed in column number 2 in the second row. This is called alignment of identical residues in columns. In addition, a dash or hyphen character is placed at position number 1 in the first row. The insertion of a dash in a sequence is called introduction or placement of a gap. The gap is introduced in one sequence whenever the residue in other sequence at corresponding position is not identical. In this way, the introduction of gaps allows the alignment of only identical residues along the sequence in successive columns. The situation of aligning a residue with a hyphen arises whenever there is an insertion of a residue in one of the two sequences. The situation of aligning a residue with a gap may also arise when there is a deletion of a residue in one of the two sequences. Now, to continue alignment of next residue in both the sequences, compare the second residue H in first sequence with third residue I in the second sequence. We find that they are not identical. So continue matching next residue of the first sequence, that is the third residue of first sequence, with third residue I in the second sequence. We find that third residue I in the first sequence is identical to the third residue I in the second sequence. Therefore, align these residues in column number 4 and insert a gap at column number 3, that is before I, in second row, which gets aligned with residue H at column number 3. Now, to continue alignment of next residue in both the sequences, we find that both residues are having fourth residue S. Therefore, align these two residues at next column in both rows. Continue with each successive position in both the sequences in the manner explained. In this process, alignment will end up as shown at the bottom of the slide. This process of lining up two sequences to achieve maximal level of identity and therefore conservation in case of amino acid sequences in successive columns is called sequence alignment. The alignment of identical residues allows the calculation of the number of identical positions. These are called identities in two sequences. Consequently, we can describe the extent of the identity numerically between two sequences. In the previous alignment, we have not allowed any mismatching of the residues in the same column. Therefore, substitution of one residue by another residue during evolution was not considered. However, in actual evolution, substitutions by similar residues are allowed because a residue may replace another residue for improved functioning. Therefore, mismatching of similar residues also need to be allowed during sequence alignment. The presence of a non-identical residue at a given position in an alignment is called a substitution. Therefore, in addition to alignment of a residue with a residue and alignment of a residue with a gap, we shall allow the alignment of mismatched residues to allow substitution during evolution. Similar residues, say amino acids with similar physicochemical properties, are more likely to replace each other during evolution. Therefore, replacement amongst similar residues such as asparagine and arginine, as well as valine and leucine, both shown in red in this slide, can be allowed because they do not disrupt the three-dimensional structure of the mutated protein to a significant extent. The substitution of a residue by such a similar residue at a position in an alignment is called a conservative substitution. The conserved substitution is not affected to affect the function of the evolving protein. Therefore, in addition to alignment of identical residues, we can allow alignment of similar residues also. However, this simplified method of aligning two sequences from left to right is not used in actual alignment because it does not result in the optimal alignment of two sequences. On the other hand, a method which guarantees to find optimal alignment of two sequences is provided by dynamic programming algorithm, and it is actually the dynamic programming which is used for creating optimal sequence alignment. In the next module on sequence alignment creation process, we'll understand the complete algorithm of dynamic programming for creating optimal sequence alignments. The alignment of identical and similar residues allows the calculation of the number of the identical and similar positions. These collectively are known as similarities. Consequently, we can describe the extent of similarity between two sequences. Similarity is a descriptive number, means a numeric score for the alignment, and implies that the two sequences by some criteria, say evolutionary, resemble each other and can be used to infer homology, that is, the two sequences have descended or originated from a common ancestor. For scoring sequence alignment, initially arbitrary scoring schemes were used. All identities, mismatches, and insertion of gaps were assigned a fixed score. The arbitrary scores were highest for identities, that is conserved positions, lower for mismatches, that is substitutions, and lowest for gaps, that is insertions or deletions. To understand comparison of different alignments, let us assign some arbitrary score for scoring the alignments. In the arbitrary way, say +2 is assigned to a column where we have identical residues matched. A lower score, say +1, is assigned for alignment of allowed substitutions, which are usually for similar physicochemical amino acids. On the other hand, whenever we have aligned a gap with a residue, we assign no score for that column, that is a 0. The total score of an alignment is defined as the sum total of all the scores of individual columns. The total alignment score is sum of scores for individual identities, similarities, and gaps. With this arbitrary scheme, the total score for this alignment will be +40 because there are 18 identical residues totaling +36, 4 conservative substitution by similar residues getting +4 score, and 26 gaps getting 0 score. The assignment of scores based on identities and similarities allows the evaluation of the alignment for its overall similarity. This score of +40 will be used later in this module while learning calculation of statistical parameter p-value which is calculated using the raw score of the alignment. Arbitrary scoring matrices are sometimes appropriate for DNA and RNA comparisons, where transitions equal transversions and the sequences are moderately diverged. However, arbitrary scoring systems are not useful for protein alignments. Therefore, many alternatives to the arbitrary scoring matrices has been suggested. One of the earliest suggestions was a scoring matrix based on the minimum number of bases that must be mutated to convert a codon for one amino acid into another amino acid was developed. This matrix, known as mutation data matrix or mutation data matrices, succeeded in identification of distant relationship among protein sequences. The mutation data matrices is an improvement, therefore, over the arbitrary scoring schemes because it incorporates the knowledge about the process of mutating one amino acid into another one. However, its use is limited because the mutation data matrices does not include the codon uses or the selection process during evolution. Therefore, in practice, practically derived, that is empirical scoring systems based on previously determined biological knowledge have been developed for alignment of one residue with another residue. In the next module, sequence alignment creation process will understand the empirical scoring systems for scoring aligned residues. An inference of homology can be derived from the identity between two sequences. The identity between two sequences is quantitative and can be used to infer homology from percentage of identical residues aligned over the length of pairwise sequence alignment. The XY graph between the number of aligned residues and percent sequence identity for known proteins show that when the sequences are homologous, they all fall in green region. Therefore, if XY graph between the number of residues aligned and percent sequence identity for a given pair of aligned proteins fall in green region, then it is safe to infer homology for these two proteins. Therefore, this zone has been named as safe zone. On the other hand, when the sequences are not homologous, then XY graph falls in the red zone. Therefore, if the XY graph of two given proteins falls in red zone, then it is not safe at all to draw an inference of homology. Therefore, this zone has been named as midnight zone. In the yellow zone, user need to be careful before inferring homology and must follow statistical assessment of sequence alignment. In the present case, with number of identities 18, which is 37.5% sequence identity over 48 residue alignment length, this alignment falls in yellow colored twilight zone. Therefore, user must follow the powerful approach of statistical evaluation using p-value to infer homology. The statistical evaluation to infer homology from alignment score is frequently assessed by calculating p-value to determine the statistical significance. The p-value is given by k multiplied by e raised to the power lambda s, where k is natural scale for the number of residues in two sequences, and lambda is natural scale for scoring system used for scoring alignment. E is an exponentiation function, and S is the actual raw score for the alignment. But the sequence alignment programs report only score. The parameters K and lambda are not reported by alignment programs. However, PRSS software available at MNet calculates the parameters K and lambda. Simply paste two sequences as plain text in the provided text boxes and click Run PRSS button. On the following page, the PRSS reported lambda equal to 0.2391 and K equal to 0.2133. Now p-value can be calculated using MS Excel. Run MS Excel and enter K, lambda, score and p-values as label in cells A to D in row 1. In the second row, enter actual values, that is 0.21, 33, 0.2391, score 40, and type the formula shown in the slide in cell D2. Now press enter. The resulting p-value is displayed in cell D2, which is approximately 10 raised to the power minus 5. To infer homology from p-value, use this table. When p-value is less than 10 raised to the power minus 100, then two sequences are exactly identical sequences. When p-value is in between 10 raised to the power minus 100 to 10 raised to the power minus 50, then the two sequences are nearly identical. When p-value is between 10 raised to the power minus 50 to 10 raised to the power minus 5, then two sequences are having clear homology. When p-value is between 10 raised to the power minus 5 and 10 raised to the power minus 1, then two sequences are having possible distant homology. And finally, when the p-value is more than 10 raised to the power minus 1, then the two sequences are just randomly related to each other. There is no homology. Since p-value in the present case is approximately 10 raised to the power minus 5, and this value falls between 10 raised to the power minus 5 to 10 raised to the power minus 1, therefore these two sequences may be having possible distant homology. The sequence alignment is represented by highlighting the conserved positions. Two ways are shown here to represent sequence alignments. The first is writing identical residues underneath the alignment using small letters or lowercase letters. The other is used by some sequence alignment programs such as emboss needle and emboss stretchers. These programs represent identities using a vertical bar and represent conservative substitutions using columns, and semi-conservative substitutions are represented using dots. These symbols are written in between the two alignments for each column. There are two types of sequence alignments. When two sequences are forced to align along their complete lengths, that is full length, then this alignment is called global alignment. In the alignment shown on the top of the slide, the middle region residues are aligned to each other involving single insertions and deletions, and end regions alignments involve insertions of large gaps involving insertion deletions of more than one amino acids. Therefore, this complete length global alignment is involving large gaps and may not be biologically relevant. On the other hand, the middle portion involving insertion deletion of single amino acids shown at the bottom of the slide may be biologically relevant alignment. Therefore, if only these residues with single gaps are considered, then this is called local alignment. That is, aligning short regions within the complete sequence length. Therefore, local alignment means aligning short regions within the complete sequence length, and these local regions represent sometimes domains, motifs, fingerprints of amino acids in actual protein sequences. The alignment between a pair of sequences is known as pairwise sequence alignment. Therefore, we have seen a pairwise global and pairwise local alignment till now. Sometimes more than two sequences are aligned in this fashion. The alignment of more than two sequences is called multiple sequence alignment, MSA. The MSA may be either global or local. We'll see the importance of local alignments in sequence similarity search and pattern matching. Dear students, in the present module, we have seen that the gene and the protein sequences are related to each other due to descent from a common ancestor. This evolutionary relationship may be inferred after aligning two sequences. However, the conclusive proof for homology is derived using statistical significance of alignment. Once sequence homology of a new gene is established with a known gene, then all the structural and functional knowledge of the known gene can be used for modeling biological processes involving new gene. Consequently, sequence alignment gained central importance in bioinformatics. Thank you for visiting EPG Pathshala.